today we'll study about chemotaxis and bacteria. Okay, so what does the word chemo means? Chemo means chemical. Okay, and taxis means movement. Okay, so the bacterial movement due to the chemical signals is known as chemotaxis. Okay, now these signals can be of two types. That is, one is positive. This is due to the attractants, and the second one is negative due to repellents. Okay, so now what happened? If you consider the bacteria, then bacteria has two types of movements. Okay, so one movement is the clockwise one. Okay, and the second one is counterclockwise. Okay, so now what's the difference between both of this? We'll see. In case of clockwise movement, this is tumbling movement. Okay, this is a tumbling movement. Okay, now in this what happened? In this particular case, the bacteria comes in contact with repellent. Okay, bacteria comes in contact with repellent. Okay, and this repellent shows negative signal. Due to this negative signal, this tumbling effect is shown. Okay, now the second one is the counterclockwise movement. This counterclockwise movement is also known as run movement. Okay, this run movement is due to the positive signal or the attractants. Okay, now this is the running effect. Okay, this is the running effect and the default effect also. Okay, and the clockwise or the tumbling movement is due to repellent, and which is due to the negative signal. Like for now you can see, so there is this bacteria and there are certain chemicals. Okay, and again here there is another bacteria. So this is negative, this is positive. Okay, if the chemical is attractant, then there is positive, it will run. Okay, and if the chemical is if the chemical is negative, then it will repel. Okay, it will repel, showing tumbling movement. Okay, then we'll see exactly how the mechanism is working. Okay, so this is the plasma membrane. Okay, this is the plasma membrane. Now in the plasma membrane of the bacteria, there is a receptor. Okay, there is a receptor that is called MCP. MCP stands for methyl accepted chemotaxis protein. Okay, MCP receptor. Now what happened? This MCP receptor is in, okay, now this MCP receptor is in dimer form. Okay, dimer form. Now say for example, a repellent comes in contact with the receptor. Okay, now this receptor is has another molecule, okay, attached to it, that is known as CHEW. Okay, now CHEW, what happens on binding? When the ligand binds to the receptor, the CHEW gets activated, and which binds to CHE. Okay, it binds to CHEA. Now, on binding to CHEA, now this CHEA get autophosphorylated. Okay, it undergoes autophosphorylation. But again, this autophosphorylation occurs only at particular amino acid, that is histidine. Okay, so this histidine only get phosphorylated. Okay, now here you can see this histidine is getting phosphorylated. So after that, what happened was the histidine histidine gets phosphorylated. Then what happened? ATP is converted into ADP. Okay, ATP is converted to ADP. Okay, now then after getting converted to ADP, now what happened? Now there is another molecule, that is CHEY. So this CHEY is a regulatory protein. Okay, is a regulatory protein, and this CHW is an adapter protein. Okay, and CHEA is sensory protein. Okay, write it here, sensor protein. Okay, now what happened? Now this CHEW, which is a regulatory protein, has a specific amino acid, that is aspartate. Okay, the symbol is D for aspartate. Okay, now the phosphorylation is transferred from CHEA to CHEY. Okay, now this CHEY get phosphorylated only at the specific amino acid, that is aspartate. Okay, after getting phosphorylated, then the phosphate group is automatically transferred from CHEA to CHEY at the aspartate amino acid position. Now what happened? This CHEY get diffused to the flagella of the bacteria. Now this flagella consists of the motor region. This motor region has a particular protein called film protein. Okay, so eventually these all are connected. So first the CHEY will get phosphate group, then it will diffuse to flagella. Then from flagella there is this motor region, and this motor region has this film protein. This film protein will help to make this clockwise movement. Okay, now this clockwise movement or the tumbling movement only lasts for 10 seconds, not for more than 10 seconds. Okay, so within this 10 seconds, now it is the signal has been given that this particular ligand is negative. Okay, it is negative, and that's why within 10 seconds it moves away from this ligand or repel from this ligand. Okay, as it is repellent, that's why it will repel. Okay, once it moves out, then again another molecule, that is CHEW, comes into action. Now this shows negative effect to CHEY. Okay, what it does now, this CHEW will remove this phosphate group from the aspartate amino acid. Okay, this will inhibit the phosphate group. Once this phosphate group is inhibited, phosphorylation is stopped, and hence CHEW cannot be diffused to flagella, and again it cannot give the clockwise movement. Okay, and by default it will go to the default state, that is counterclockwise movement or the running movement. Okay, so this is all about the negative signal. Okay, negative signal. Now coming to the positive signal, in case 2, that is positive signal. In case of positive signal, same. Now you have this same receptor. You have MCP over here, and then same CHW and CHA. Now what happened in case of when now if you have this ligand, okay, which is attractant, now it will give a positive signal that this is an attractant. Okay, in this case what will happen in case of chemoattractants, when a ligand binds to MCP, then this receptor, what happens, brings another protein, that is CHER. Okay, now this CHER methylates the CHEW. Both the here it will methylate both the side. Here it will methylate. This side also it will be methylated. Okay, the methylation restrict the phosphorylation of CHEA. Okay, so the phosphorylation is restricted. So if the phosphorylation is restricted, then the further process which we understood in the negative signal will be inhibited. Okay, and hence the running movement will take place. Running movement will take place. This is the default movement of bacteria. Okay, so this is all about the movement in bacteria or the chemotaxis. Okay, thank you.